am Ziheng from Department of Chemistry. Dalhousie University. Today I'm going to introduce ultraviolet photodissociation. Good morning, everyone. I am Ziheng from Department of Chemistry, Dalhousie University. Today I'm going to introduce ultraviolet photodissociation, UVPD technology, and some of its application for top-down proteomics. I am a first-year master student in Dr. Alan Doucet's lab, and my research is about protein sample processing ahead of mass spec analysis. For this class, I was willing to present something different from my projects, and although I'm not doing top-down proteomics now, I'm interested in it, especially the new technologies related to it. That's why I choose UVPD as my topic, because it is a relatively new technology for proteomics. It is developing and seemingly has broad application prospects. Within the last few decades, scientists have discovered that the human proteome is much more complex than the human genome. It is estimated that the human genome comprises between 20 to 25,000 genes. The total number of protein in the human proteome is estimated as over 1 million. Why? Well, first, single genes can encode multiple proteins. Second, protein has post-transitional modifications. There are chemical modifications on the protein, and they play a key role in functional proteomics because they regulate activity, localization, and interaction with other cellular molecules such as proteins, nucleic acids, lipids, and cofactors. There are several types of PTMs. Not only these, there are more. And the most important ones are glycosylation, phosphorylation, acetylation, and methylation. These PTMs can dynamically alter the structure, stability, and function of protein in the cells. The determination of proteins and their PTMs is particularly important for the study of heart disease, cancer, neurodegenerative disease, and diabetes. When we study proteomics, we do need to be able to identify these PTMs. As we know, until now, bottom-up strategy is still the dominant strategy for proteomics. It has been popular because it's easier to identify peptides because they are smaller. There will be a huge number of peptides in one sample, but the spectrums won't be too complicated for each one. However, it is hard to identify the original intact proteins. What if there are proteins isoforms? What if the protein has some PTMs on some non-specific peptides? All this information will be lost. The sequence coverage of mainstream technology based on bottom-up strategy is low, mostly less than 20%. That means more than 80% of the protein sequence remains unidentified. So I would see bottom-up as a compromise when the technology is not advanced enough. With the development of mass spectrometry, exactly the top-down proteomics approach has become more and more popular. It has unique advantages for the identification and localization of PTMs and sequence variations. For top-down, protein identification in a real sense can be achieved through the quality of the complete protein and its fragmentation spectrum information, and its sequence coverage can reach 100%. That is to say, top-down can retain the association information between various PTMs. The most critical steps of top-down proteomics are separation, mass spectrometry, and analyzation, and data processing. For mass spec part, we have already known that for top-down proteomics, we need the instrument with extremely high resolution to measure the molecular weight of the protein, because the protein ions are always multi-charged. Mostly we use FT-ICR and Orbitrap, but here I'm going to discuss another part, fragmentation. Fragmentation means that we need to break the chemical bonds. That means some energy more than the bond dissociation energy will be needed. For protein or peptide backbones, we have N2C and C2C bonds. The average bond energy of these two are both around 3 to 4 according to the literature, but they are average values. Of course, for protein, bond energy is affected by the groups on the side chain, so it varies. Someone has calculated the energy of amide bonds. It's generally around about 3.2 to 4.5 electron volts. And generally, for protein backbones, the strongest bond is usually C to C, that is C alpha to CO, then N to C alpha. The weakest bond usually is N to C, that is the peptide bond. So we will find something have more energy than this to break the bonds, that is to fragment the protein ions. Well, let's briefly go over the main fragmentation methods. There are mainly 3 types of fragmentation methods or technologies. First, collision-based. It's CID and HCD. And second, chemistry-based. It's ECD and ETD. And then photodissociation. It includes IRMPD and UVPD. All these methods use some particle to hit the ions to fragment them. Of course, the particles bring energy with them. When they hit the precursor ions, they transfer energy to the ions in some way, and thus break the bonds. Collision-induced dissociation, CID, is a classical and also the most popular fragmentation technique. It's also widely applied on proteomics. For CID, select precursor ions are accelerated by an electrical potential and then allowed to enter the collision cell or ion trap where the collision takes place. Then the precursor ion collide with neutron molecules of the collision gas, often helium, nitrogen, or argon, in the collision cell. Some of the kinetic energy is converted into internal energy of the precursor ion. The precursor ion undergoes repeated collision with the collision gas, building up potential energy in the molecule until eventually the fragmentation threshold is reached and the precursor ion fragments into product ions. Now, when we talk about CID, we often talk about 3 types of it. They are low energy CID, high energy CID, and HCD. Low energy CID is typically carried out with ion kinetic energies less than about 1 kilo-electron-volt. For low energy CID, fragmentation reactions are often limited to neutron losses depending on the nature of the precursor ions. The neutron losses could be water, methanol, carbon monoxide, carbon dioxide, acetonitrile, ammonia, and so on. These neutron losses are often not very helpful for us to identify the structure of the molecule, although sometimes They can be used to obtain information about functional groups. Low-energy CID takes place in quadrupole, ion traps, and FTICR, where the kinetic energy of ions cannot be too high. The second type is high-energy CID, also named HE-CID. When the precursor have higher energies, between 1 to 20 kiloelectronvolts, once the collision happens, the carbon scalene of the molecule can be broken. These are much more structurally significant and often result in cleavage of the molecule at characteristic positions. For intact protein, HE-CID perform better than low-energy CID, generating more informative fragments. However, if the energy is too high, uncontrolled fragmentation can occur. This should be avoided. High-energy CID only can take place in magnetic sectors and TOFs because ions can be accelerated to very high kinetic energy only in these instruments. So the application of HE-CID to top-down proteomics is limited because the resolution of TOFs is not as good as FTICR and Orbitraps. So how does CID perform with proteins? CID can fragment peptides and small proteins well. Typically, it breaks amide bonds to obtain B and Y fragments. Amide bond is the weakest bond on the backbone of the protein. CID also can break some side chain, cut off some neutron loss. CID does have advantages. It works well with all kinds of mass analyzer. Of course, the energy is different according to the analyzer, but there are enough choices. CID also has pitfalls. First, in the collision cell, under the RF-only mode, some lightest ions will inevitably hit the wall and be lost. It means that the CID cannot detect all of the ions. It is called low mass cutoff. Second, for intact proteins, CID often has problem to fragment the ions well. Well, an example. Here is an argon in the collision cell, and we let a 1 kD peptide ion with 30 electron volt of kinetic energy move forward and hit the argon. A part of the kinetic energy is transferred to internal energy of the peptide ion. According to the literature, only 3.8% of the initial ion energy, which is 1.2 electron volt, is available in the center of the mass frame for internal excitation and subsequent fragmentation of the ion. 1.2 electron volt is less than most covalent bond dissociation energies, so it's impossible to get a bond cleavage, a direct bond cleavage, at least in the center. In fact, CID tend to fragment more efficiently at protein termini or near charged residues, providing incomplete sequence coverage. If we use higher voltage to accelerate the ions more, it will be better. But 1 kD is even not an intact protein. If we have a larger protein like 50 kD, so this will always be a problem for intact proteins. Higher energy C-trap dissociation, HCD, is a new kind of CID. It was developed to extend the range of CID methods. There are high RF voltage in the C-trap to retain fragment ions. The HCD cell is used to fragment the ions, after which they are accelerated into cool down and stored inside of the C-trap. The kinetic energy of the ions are higher than traditional CID, so it can fragment better than CID. HCD does not have limitation imposed by a RF trapping field, so unlike conventional CID, HCD does not have a lower mass cutoff. Better fragmentation, no lower mass cutoff. These two advantages has made HCD a robust tandem mass strategy for quantitative proteomics. Now I'm going to introduce photodissociation technology. Unlike CID, photodissociation introduces photons to excite ions and kind of heat it to get fragmentation. For photodissociation, we use laser to get enough photon energy. Laser stands for light amplification by stimulated emission of radiation. And how do we get laser? There's a lot of theoretical thing in it. We only need to know that we introduce some photons to some gain medium, and the atom of the gain medium absorbs energy, which raises some electrons into excited quantum states. Then it tends to emit photons to go back to the ground state energy level. When the number of particles in one excited state exceeds the number of particles in some lower energy state, the rate of stimulated emission is larger than the rate of absorption of light in the medium, and therefore the light is amplified. But usually, we need an optical cavity, a pair of mirrors on either end of the gain medium. Light bounces back and forth between the mirrors, passing through the gain medium and being amplified each time. Typically, one of the two mirrors is partially transparent. Some of the light escapes through the mirror. This device is sometimes called a laser oscillator. Lasers are classified into 4 types based on the type of gain medium used. There's solid state, gas, liquid, or semiconductor. This picture is all of the commercially available lasers, and different color means different gain medium types, and the name of the gain mediums are all labeled for each laser. You can see in the left part in the UV zone, we got several lasers which have already applied for UVPD, and on the right end we got an infrared laser with CO2 gain medium. This is used for IRMPD. IRMPD, okay. This is short for infrared multiphoton dissociation technique. For IRMPD, we use CO2 laser to heat apparent ion. But due to the relatively low energy of IR photons, which is about 0.1 electrovolt, multiple photons are needed to accumulate energy to reach more energetic vibrational state until the bond is broken. IRMPD is most commonly used in FTICR mass spectrometry. In biomolecules, the CCCN and PO bonds can effectively absorb IR photons to get dissociated. In the structural analysis of biomolecules, IRMPD is mostly used for the structural analysis of phosphorus-containing polypeptides. Now it's time for ultraviolet photodissociation, UVPD. Similar as IRMPD, for UVPD we use UV laser to heat apparent ion to get a bond cleavage. The difference is UV laser have more energy than IR laser, so we won't need multiphotons to break a bond. And there must be some difference in the fragmentation pattern. Before we discuss the differences, let's see a chart first. I searched some keywords on Google Scholar. The blue bars are UVPD together with top-down proteomics, and the orange bars are UVPD together with mass spectrometry. We can see that both have grown rapidly in the past decade. So, why had UVPD become so popular? Let's get some points first by comparing UVPD energy diagram to CID and IRMPD. This energy diagram illustrating energy deposition by collisional activation or absorption of IR or UV photons. M plus represents a selected precursor ion, and Fn plus represent various fragment ions with different activation energies. Because of the stepwise nature of energy deposition, both IRMPD and CID to promote access to lower energy fragmentation pathways depending on the rate of ion activation. In short, the fragmentation pathways with the highest activation energies, like the F1 plus, F5 plus, are not accessible if the precursor ions dissociate via other lower energy channels. During the energization process, UV photons, however, have much greater energies, and thus absorption of even a single UV photon, for example, 193 nanometer photon have 6.4 electron volt energy. It can populate excited electronic states that afford access to fragmentation pathways with significantly higher activation energies. The nature of UVPD result in the unique fragmentation patterns. Next, I'm going to introduce UVPD by its applications. An UV laser was first coupled to a mass spectrometer for photodissociation over 3 decades ago, in 1979. In 1984, William D. Bowers and his colleagues reported that oligopeptide ions stored in an ICR analyzer cell are fragmented efficiently by UV laser radiation. He found that 193 nanometer works best for fragmentation, and it breaks C2CR bonds and also CO2NH bonds. After that, more and more studies of UVPD for biomolecules had been published. You can read this 2009 review to get more idea about that. People tried different UV wavelengths. People coupled UVPD to different mass analyzer, FTICR, TOF, and so on. And then, in 2013, Broadbelt reported an application of UVPD on intact protein. Dr. Jennifer S. Broadbelt is the person who invented the commercial instrument with an optional part of UVPD and applied it to intact proteins. She is a professor of chemistry at University of Texas at Austin. Her group focuses on the development and applications of photodissociation mass spectrometry for studying biological molecules, not only for peptides, proteins, but also nucleic acids, lipids, and so on. Dr. Broadbelt has worked with Thermo Fisher Scientific to implement the technique on Orbitrap instruments. Let's watch a video to study how the UVPD works with an Orbitrap. This is a novel Orbitrap IQX trip-rate mass spectrometer with UVPD source. Here is the UVPD source. It generates laser pulses and then goes into the collision cell through two specular reflections. When we do mass spec, ions go through the RF lens here and then cross through the advanced active ion beam guide for preventing neutrons and high-speed clusters from entering the next quadrupole. This is a quadrupole mass filter. One or more precursor ions are selected here. And then ions goes into the CID collision cell. Here they can be fragmented by a low energy CID. After this, ions go into C-trap. C-trap is a curved linear trap. By rapidly ramping down trapping RF voltages and applying DC gradients across the C-trap, ions can be bunched into short packets. Then the ions go into the HCD cell. Here, the laser hits the ions and generate the fragment ions. Ions then go back to C-trap. There's nitrogen gas in the C-trap. Ions collide with nitrogen and rapidly lose kinetic energy. This is called cooling down. At last, ions are suddenly ejected into an orbit trap and to be detected there. In this instrument, you can do CID, HCD, and UVPD, and you also can combine HCD with UVPD.